So as Jerry mentioned, I've spent my career working on the ryanodine receptor, which is shown here. This is a three-dimensional reconstruction of a high-resolution image. I'll show you more. And it's the largest ion channel. And what I'm showing you here is a great white shark that I photographed off of Guadalupe Island off of the coast of Mexico last year. And I'm showing that the shark is now eating the ryanodine receptor. And that's just to let you know that this is a big channel. It takes a big fish to eat it. And I'm going to talk about the structure, the biology of the channel as it relates to different organs. And I want to mention disclosure that Columbia has a number of patents on drugs that I'll talk about that target the ryanodine receptor, fix a leak, and are in clinical development now. And so I have a commercial interest in this company and want to just acknowledge that there's a potential conflict of interest before moving on. So ryanodine receptors, as Jerry mentioned, are intracellular calcium release channels. They're present in virtually all types of organs, all tissues. And depending on which organ they're in, they subserve different cellular functions. They're involved in neurotransmitter release in the brain, and in heart and skeletal muscle, excitation-contraction coupling, and in pancreatic beta cells involved in glucose metabolism. They are homotetrameric channels, and they sit on intracellular membrane, either sarcoplasmic or endoplasmic reticuli, and they're tetramers with 4 identical protomers. And this little blue molecule here, you'll hear more about that, is called FKBP12 or calstabin, and it stabilizes the closed state of the channel, and under pathologic conditions that are caused by stress, oxidation, phosphorylation, it can be dissociated from the channel, enabling a leak of calcium, which can be pathologic. And so if you look at the cartoon of a muscle cell, it could be cardiac or skeletal muscle, there are a couple of features which are unique to striated muscle. So there's a plasma membrane which invaginates into the muscle and has a specialized function called the transverse tubule. On that membrane are voltage-gated calcium channels. So when the membrane is electrically excited by depolarization, they in turn activate much larger ryanodine receptors on the sarcoplasmic reticuli, and they release calcium, as Jerry was mentioning. And this is the basis for muscle contraction in your heart and in your limbs. When I was cloning the ryanodine receptor in the late 1980s, I was the only person I knew who could actually see what he or she were cloning, because the ryanodine receptors are so large that in this beautiful electron micrograph by Clara Franzini Armstrong, you can see individual ryanodine receptor complexes spanning between the transverse tubule and the terminal cisternae of the sarcoplasmic reticulum. So each one of these arrows represents a single channel seen with tannic acid negative staining. And so we've learned quite a bit more about the ryanodine receptor over the last 30 years or so. And as I mentioned, it's a homotetramer. It actually is the largest known ion channel. The complex has a molecular mass exceeding 3 million Daltons. Most of the sequence encodes this enormous cytoplasmic domain, and here's the transmembrane domain sitting in the sarcoplasmic reticulum or endoplasmic reticulum. The channel is a macromolecular signaling complex, so it has kinases, phosphatases, phosphodiesterases, and other regulatory proteins targeted directly to the channel through sequences that have been conserved from C. elegans to humans. So there's an enormous amount of evolutionary conservation. In the heart and in skeletal muscle, there's a voltage-gated channel on the opposing membrane called the transverse tubule, as I mentioned. And when this membrane is depolarized, the voltage-gated channel is activated. In the heart, a small amount of calcium enters, binds to and activates the ryanodine receptor to release calcium, bind to troponin C, allowing actin-myosin cross-bridging and sarcomere shortening, which is the basis for muscle contraction. In skeletal muscle, there's believed to be a direct interaction between the voltage-gated channel and the ryanodine receptor, which leads to gating of the ryanodine receptor upon depolarization of the plasma membrane. Using cryogenic electron microscopy, which I'll tell you more about in a few moments, we were able to deduce a high-resolution structure, as Jerry mentioned, at about 4.5, 4.8 angstroms. Excuse me. I'll show you more about this. It was published last year in January, and in that same issue, of Nature, there were 2 other groups, so 3 articles published on the ryanodine receptor, all showing the high-resolution structure, one headed by Ning Yan at Tsinghua University and one by Efremov at MRC. And so I guess you could say that it took 3 articles to elucidate the structure of such a big channel. We were able to map each one of the protomers, and shown here in color is one of the 4 protomers, and then the rectangle above shows the different regulatory domains of the channel, which had been hinted at from functional studies, but this was the first time that they had actually been mapped structurally. So it's interesting, I think, to look back historically to where our understanding of excitation-contraction coupling comes from. And one of the first giants in the field is Antony van Leeuwenhoek, who was using this home-built microscope and drew these remarkable figures of striated muscle. And this is where the term striated muscle came from. And he shows the relationship of the membranes that I was telling you about. If you fast forward about a century to Luigi Galvani, you begin to appreciate a large dose of serendipity or luck in this field of excitation-contraction coupling. So Galvani was working with a galvanometer producing electrical energy and the hind limbs of a frog, and wrote in his lab notebook, while one of those who was assisting me, so this is blaming it on his technician, touched lightly and by chance the point of his scalpel to the internal crural nerves of the frog, suddenly all the muscles of its limbs were seen to be so contracted they seemed to have fallen into tonic convulsions. This is believed to be the very first observation of the connection between electrical energy or electricity and muscle contraction. And prior to this observation by Galvani, it was an absolute complete mystery of how our muscles contracted, what made them contract. So fast forward another century or so to the famous British physiologist Sidney Ringer. And Ringer was using a beaker full of solution with a heart in it and adding various hormones and seeing what the effects were. And he wrote in his lab notebook, after the publication of a paper in the Journal of Physiology in 1883, I discovered that the saline which I had used had not been prepared with distilled water, but with pipe water supplied by the New River Water Company. As this water contains minute traces of various inorganic substances, I at once tested the action of saline solution made with distilled water and found I did not get the effects described in the paper referred to. Well, obviously, this is every scientist's worst nightmare of not being able to reproduce your own results, especially after they've been published. But it led to the remarkably critical discovery that what was missing from the distilled water was, in fact, calcium. And when Ringer added back calcium, everything worked beautifully. So this brought to the full circle the role of the membranes, the role of electricity, electrical energy, and the role of calcium. So my own contribution to this field also has a large dose of serendipity. In the late 1980s, I was at Harvard, as Jerry had mentioned, and collaborating with Sidney Fleischer, who was a great biochemist for membrane proteins, and Sid had purified ryanodine receptor, sent it to the lab I was working in at Children's Hospital, Bernardo Nedelgenard's lab, and I was given the protein and said, clone it. And so I ran on a protein gel, and we had absolutely no idea how big this thing was because the molecular weight markers in those days stopped at 97 kilodaltons. So I collaborated with Paul Temps, who was then in Phil Leader's lab, and was an excellent protein biochemist. And we proteolyzed the ryanodine receptor. HPLC purified these peptides, and I used that to design probes for cDNA cloning. After cloning the channel, discovered that one of the peptides, we then called KC7, was not part of the enormous ryanodine receptor, 560,000 Dalton protein, protomer, but was a then novel protein that migrated much at the bottom of the gel, and clearly we never would have noticed it had it not been for the serendipitous sequencing of its amino terminus. We gave it the name calstabin for calcium channel stabilizing binding protein, and as I'll show you, it's an integral component of the ryanodine receptor macromolecular complex. Well, what is this protein? Well, it has another name called the FK506 binding protein, or FKBP12, and it was first discovered as a cytosolic receptor for the immunosuppressant drugs FK506 and rapamycin. And work in my laboratory in the mid-1990s, I think Jerry mentioned, I'm also a cardiologist, was interested in the problem of stent restenosis. So in the mid-90s, cardiologists began putting in these metal stents, like this one shown here in the upper right, to keep arteries open and prevent heart attacks. And routinely, about 6 months after the insertion of the stent, about half the patients were coming back with the stents filled up like this. I went down to the pathology department and took a look at that, and it was just beautiful sheets of smooth muscle cells, not atherosclerosis. And we showed in the laboratory that rapamycin binding to FKBP12 inhibits vascular smooth muscle cell proliferation and migration. And then went to J&J, which owned a company at that point making stents, and they were searching for something to put on the stent. And we showed in this pig model that we could prevent intimal proliferation, and they managed to coat the stent with rapamycin, and it's now marketed as the Cypher stent. And it's reduced the incidence of restenosis from about 50% to less than 10%. Now, remarkably, this identical same molecule, which we call calstabin, has a completely different function, which is its natural cellular function, which is to stabilize the closed state of the ryanodine receptor and prevent a pathologic leak of calcium. One of the early clues to what the calstabin or FKBP12 was doing in the channel was this experiment that took recombinant ryanodine receptor, expressed it in insect cells which lacked calstabin, and reconstituted the recombinant channels into a planar lipid bilayer, recording currents through single ryanodine receptor channels. And in panel A, you can see very chaotic behavior of the channel in the absence of the stabilizing subunit. And then when co-expressed with calstabin, you get these beautiful, long, stable closed states. Later on, when we knocked out the calstabin in mice, they had leaky channels in vivo. And from these types of experiments, both in vitro and in vivo, came the concept that the calstabin prevented a leak in the channel. Well, about 12 years ago, I think Jerry was still dean then, I was asked to give a dean's lecture. And afterwards, Wayne Hendrickson, who many of you know is one of the world's great structural biologists, came up to me and said, "Andy, would you like to work on the structure of the ryanodine receptor with me?" And I thought, of course, this is amazing. So we had been purifying ryanodine receptor for many years, and we had a pretty good protein prep. And we readily got crystals, and they diffracted to about 7 angstroms. And this is what the electron density map of one of those crystals looks like. Here's an individual ryanodine receptor, and then every other channel is facing away in the crystal. And this was phased using the envelope of the then available 3D Cryo-EM reconstruction. So being very naive about structural biology then, and I probably still am now, I thought, wow, we got crystals, we're on our way. And 12 years later, we still had 7 angstrom crystals. And then people in this room know way better than I do, a revolution occurred in Cryo-EM, and we were attracted to that. And with the availability of a great team at Columbia, we were able to, within about 6 months, solve the high-resolution structure that we had been eluding us for over a decade. And so here's a team, Wayne Hendrickson and I had been working on this. Ron Zalk had been in my lab now for 10 years working on this project, and had gotten to the point where he was now coming into the lab and surreptitiously looking at things like soccer on the internet and so on, anything but to try to crystallize the ryanodine receptor. But he developed a great ability in purifying the protein. Joachim Frank, who many of you know is one of the fathers of Cryo-EM, also at Columbia. And Wayne and I share a postdoc, remarkably talented crystallographer and modeler, Ali Clark. Amadei DeGeorge in the back was in Joachim's lab, did the Cryo-EM, and Steve Ryken, Filippo Mancia, and Bob Grasucci helped out with technical aspects of the project. We're seen here now having imbibed 3 bottles of champagne after finding out that the paper was finally accepted for publication. So everybody's looking pretty happy. Huh? There was actually 6 bottles, no. So I'm not going to go through all of this. I think with Bridget and Clint here, it would be much better to hear from them about Cryo-EM, but in a very simplistic terms, the reason that it's so useful for such a large molecule as the ryanodine receptor and for ion channels in general is because, first of all, we use a very small amount of protein. You freeze it rapidly in liquid ethane on a grid, either carbon or now gold grid, and a very thin sheet of ice is formed. And your molecule of interest, individual single particles, is arrayed in a random orientation. And based on that, a three-dimensional reconstruction can be obtained. And for ion channels, this is particularly useful because if you think about the way we think of ion channels is really in terms of probability. So you have a preparation where the open probability is 50%, for example, which is very high for an ion channel. It means half the molecules are going to be open, half will be closed, in a sense. And so there's a lot of heterogeneity. In addition, we find out from these studies that there's a huge amount of flexibility in the cytosolic domain of the channel, probably explaining our difficulty in getting high-resolution crystals. We had, however, been purifying ryanodine receptor, and we could do that pretty well for many years. And shown here, we had a affinity column using that calstabin subunit size exclusion column, and we also knew that the phosphorylation status of the molecule was very important for determining its activity, and so we dephosphorylated it and showed at a number of different sites that it was in fact dephosphorylated. And that was the preparation we used for Cryo-EM. Another approach that proved to be very important was to do particle classification. So you can see that the single particles of the channel, some of them are very complete, like this 1,5 here, but others are degraded, and we're able to exclude those from the analysis and still have enough data left to generate a beautiful three-dimensional reconstruction. One of the classes of particles shown here had 3 of the 4 protomers, and based on that, we were able to map the outline of the individual protomers with good accuracy. So what came out in that article published about a year ago, along with the 2 other groups, all 3 groups had remarkably similar models, which gave us great comfort, was that the ryanodine receptor was for the first time identified as a member of the 6 transmembrane cation family of ion channels. And we were able to map much of the cytosolic domain. One of the protomers is shown here, and identified that most of it was comprised of alpha solenoid repeats, which form a very flexible scaffold for regulatory proteins of the channel. A couple of interesting features are an S6 segment protruding up from the membrane, interacting intimately with the C-terminus of the channel, which, as I'll show you, becomes important in our model of how the channel gates from the closed to the open state. We also identified where the calstabin, or FKBP12, that yellow triangle bound, as well as multiple other regulatory domains, including phosphorylation sites on the channel. And what was quite remarkable was that when you compared the pore domain to that of a voltage-gated potassium-sodium channel at TRPV1, you can see in each case the color is the other channel and the gray is the ryanodine receptor, remarkable evolutionary conservation of the overall structure. One of the puzzling features, though, is that the conductance of the ryanodine receptor is enormous. It's about 100 picosiemens for calcium and over 600 picosiemens for potassium, easily more than 10-fold that of a voltage-gated channel. And yet the structure of the pore looks very similar. However, what I'll draw your attention to is the selectivity filters in the other channels, which are not present in the ryanodine receptor. So these sequences here provide selectivity. Ryanodine receptors are only 11-to-1 selective for cations over anions. There are also an enormous clustering of negative charges in the lumen of the sarcoplasmic reticulum, which obviously could serve to elevate the concentration of calcium at the mouth of the channel so that when the pore does gate open, there would be a ready flood of calcium. Another interesting feature that emerged from the structure was this bend in the S6 segment, which is due to a glycine residue right in the middle of the segment that gives a 24-degree bend, which again turns out to be an interesting feature involved in the gating of the channel. So the ryanodine receptors, as I mentioned, are not voltage-gated, and yet they share a lot of structural similarity with voltage-gated channels. And what's really remarkable is that although the overall topography is conserved with S1, 2, 3, and 4 segments forming what we call a pseudo-voltage sensor, because unlike the voltage-gated channels in which the S4 segment has a clustering of every third residue is positively charged. In the ryanodine receptor, there's only a single positive charge there. So through evolution, the channel is either lost or never had the ability to be gated by voltage. Looking at the pore domain, you also again draw your attention to this extension of the S6 segment, which now can be seen to be interacting intimately with the cytoplasmic domain of the channel. So when we published that structure back a year ago, the overall resolution was about 4.8 angstroms. And readily within the past year, we've been able to improve that quite dramatically. Now, again, depending on which portion of the channel you look at, down to below 4 angstroms. And ironically, the highest resolution in this resolution map is in the pore segment that goes through the membrane, and much less the resolution is much less in the outer portions of this cytoplasmic domain. And again, this likely explains why we had difficulty in getting high resolution crystals. But you can see that we can see now the side chains of all of the resolvable portions of the channel. And depending on how we mask the res map, you can see that we can get up to about 3.6 angstroms for that core, membrane core of the channel. So this shows a tracing of the alpha backbone of the chains of the channel. And I think we've now pretty much resolved all of the ordered sequences in the channel. And again, the resolution is very nice. And as I'll show you in a moment, we're able to see ligands that gate the channel. And it's really quite remarkable. So the ryanodine receptor, why is it called this? Ryanodine is a plant alkaloid that's found in South American plant called Rhyania speciosa. And South American natives put the ryanodine on the tip of their blow darts because it locks the channel in an open configuration, and it's actually a subconductant state. So here's fully open, and ryanodine locks the channel in a half open state. When your ryanodine receptor is locked open, calcium floods out, your muscles go into tetany, your heart stops beating, you can't breathe, and your limbs, you can't move. It's a terrific insecticide. When an insect takes a bite of this leaf, it's paralyzed and falls off. And DuPont has developed a derivative of ryanodine, which is 1,000-fold more potent than DTT. And so, of course, we were interested to understand how ryanodine locks the channel in this half-open state. And we were able to visualize ryanodine molecules present in the pore of the channel in this difference map compared to with and without ryanodine. And when you trace the dimensions of the pore going through the channel, this is the tightest constriction here. These conditions here are with high concentration of EGTA, so very low calcium, which maintains the channel in a closed state. Ryanodine, shown here in green, only binds to the open state of the channel. And what we've discovered from the structural data is that when the first ryanodine molecule binds to the channel, through steric hindrance, it inhibits other ryanodine molecules from entering to the pore, and it doesn't block the channel. This is only one of these, and so this explains the partial conductance of the ryanodine modified channel. Then as you increase the concentration of ryanodine, you can force additional molecules into the pore and block the channel. Prior to these studies, the hypothesis was that there was distinct high affinity and low affinity ryanodine binding sites, and we think, in fact, that all of the ryanodine binding occurs here, right in the pore. So we've been able to solve both the closed state, which we published last year, and now an open state of the channel, and can compare the pore structure in now the closed and open state. And the red denotes the tightest constriction point, and that turns out to be only large enough for a single hydrated calcium ion to pass at a time. Again, this was a surprising finding given the enormous conductance of the channel. The opening and closing of the channel is associated with major structural changes in the cytoplasmic domain, reflected here, going from the closed to the open state. And we're in the process now of trying to understand in a very detailed way how those changes in the cytoplasmic domain determine the gating of the channel. This is the view you would see if you were a calcium ion in the sarcoplasmic reticulum, and you can see that go from the closed state where the pore is about 5 angstroms, you go to just over 10 angstroms, and that's the pathway for calcium. Now, quite remarkably, with our recent higher resolution structures, we've been able to identify a novel calcium binding site. We've been able to see where ATP binds using difference maps, and a potent regulator of the channel, caffeine, an activator. So these single channel tracings, for example, show the effects of activation of the channel, say with ATP, going from an open probability of 0.004, approximately 100-fold increase to 0.4, or caffeine, another potent activator of the channel. And when we morph from the open to the closed state, as shown here in this movie, you can see a straightening of this S6 domain, which again is embedded in the C-terminus of the channel. And fascinatingly, these activating ligands surround the connection between the S6 segment and the cytoplasmic domain of the channel. And we believe they rigidify critical interfaces that allow the binding of calcium now to be much more efficient in terms of opening the channel. So if you have calcium alone at an optimal concentration, and this curve over here shows the bell-shaped response of the channel to calcium, and this is from a 1991 paper by Barbara Ehrlich and Ilya Bezprezvani. And what they showed was that at micromolar calcium, the channel has its highest activity level. But if you look closely, this turns out to be normalized activity, and the actual open probability of the calcium-modified channel alone is only about 10%. And now when you add ATP and caffeine, you rigidify this interaction and make the binding of calcium much more efficient. So over the last 15 or 20 years or so, we've been able to study the regulation of the ryanodine receptor in different disease states. The first one we published was in 2000, looking at heart failure, and heart failure is a chronic state of stress. Patients have very high catecholamine levels, and what we showed was that the ryanodine receptor in failing hearts becomes heavily phosphorylated and oxidized, and this prevents the tight binding of the stabilizing subunit calstabin, causing a leak of calcium that can promote heart failure or cardiac dysfunction and arrhythmias. In skeletal muscle, we made similar findings in models of muscular dystrophy and in age-dependent loss of muscle function. About 50% of us, when we reach the age of 80, will develop muscle weakness that predisposes to other problems. And recently, we were able to show in pancreatic beta cells that ryanodine receptors with mutations that make them leaky can impair insulin secretion. And remarkably, we had mouse models for human mutations, and we went and looked at 27 patients with leaky ryanodine receptor mutations. And these were young, healthy people who had no suspected diabetes, and all of them were found to have impaired insulin secretion and glucose intolerance. So the basic story that's emerged from these studies and others is that stress induces a leak through the ryanodine receptor, a leak of intracellular calcium, and it's related to the loss of the tight binding of this stabilizing subunit. So this lent itself to a very obvious potential therapeutic approach, which would be to figure out a way to prevent the loss of the stabilizing subunit from the channel. And in looking in the literature, I found this molecule, JTV519, which is a 1,4-benzothiazepine made by Japan Tobacco when they were making the voltage-gated calcium channel diltiazem, which is a commonly used drug. But this molecule had no purpose, no use at that time, and no known mechanism or target. However, there have been about a half a dozen papers published, including one by a Japanese group at Yamaguchi University, showing that it was cardioprotective and improved cardiac function in heart failure model, and that it was related somehow to disturbances of intracellular calcium. So in collaboration with chemists at Columbia, I asked them to synthesize this, and in looking through the literature, found out that this JTV519 is not water soluble, and it's a potent hERG potassium channel blocker. hERG potassium channel blockers cause sudden death, and it's one of the screening criteria that pharmaceutical industry uses to eliminate molecules with potential toxicity. So looking at the literature, I thought that maybe these two rings over here seem to be similar to other hERG blockers. And so I asked the chemists to cut those off, and they said, Andy, you're an idiot. You have no idea what you're talking about. That's not how you do medicinal chemistry. And I'm sure they're right. So I looked at that and I said, well, just give me that guy there. And that turned out to be very similar to this molecule. And what's shown here is when you take the oxidized phosphorylated channel, mix it with a stabilizing subunit, it doesn't bind, and then you put in these different compounds and now do an immunoprecipitation in this case, and you can see good binding, which we know correlates with fixing the leak in the channel. And so that drug, which we now call Rycal, binds to this green peptide here, we think, using ligand binding studies, although this is very preliminary. But what's of interest is that sequence, which may bind the Rycal, is immediately adjacent to the stabilizing subunit calstabin. And here is a structure of one of the molecules I'll be talking about. We call the class of drugs Rycals. This one in particular is called S107, and it's the one I've used in the laboratory. It has a very short half-life, was felt not to be an ideal clinical candidate, but we just put it in the drinking water and give it to mice, and I'll show you some data based on that. In any case, it's intriguing, and again, this needs a lot more work, that the binding site for the drug may actually be in the same part of the channel as the stabilizing subunit, which is involved in its mechanism of action. So because of the Simon Foundation interest in the brain, I thought I would include a brief mention of some work that we have done on the neuronal forms of the ryanodine receptors. And so we published this paper now several years ago, and this cover actually was the artwork from my Israeli postdoc's wife. And when they showed me this and said, let's send this to Cell, I laughed at them. I said, they'll never publish this. And in any case, what we showed in this study was that leaky neuronal ryanodine receptors are linked to stress-induced cognitive dysfunction in a rodent model of post-traumatic stress disorder. Well, PTSD, as you know, is an extremely important problem, and here's an example of the Fukushima nuclear disaster, and these are photographs taken by a colleague of mine, Alexis Dudden, who's a history professor at UConn, who specializes in this problem. And this is a gentleman living in these temporary shelters in the heavily radiation poisoned area in northern Japan. This sign, which Alexis translated for me, says, sorry to cause any inconvenience, decontamination work in progress. And that shovel shown below is actually at a schoolyard, and these are some workers who are working to decontaminate the radiation while kids are still going to school. So this is another form of post-traumatic stress disorder, as you can imagine, as well as in countries like Israel and South Korea that live constantly under the threat of their bellicose neighbors sending nuclear weapons in their direction. In any case, having done this study and discovered that stress does indeed cause leak in neuronal ryanodine receptors, I became very interested in the problem of Alzheimer's disease because, as many of you know much better than I, there's been a long history of connection between calcium and Alzheimer's disease. And in fact, Mike Berridge has written several reviews, the titles of which are The Calcium Hypothesis of Alzheimer's Disease. So interested in that, I called Mike Shalansky, our recently stepped down chair of pathology, who for many decades has studied Alzheimer's. And I said to Mike, I'd like to get some samples of frozen mouse brain from models of Alzheimer's disease. And he said he'd be glad to send me those, but he also said, why don't you look at some of the human samples we have? And so we did, and we got 3 sets of samples, and these are just some examples, but there was an N of 5 to 10 in each one. So he had something that he called super controls, which are age-matched human brain samples from individuals who have absolutely no age-dependent neuropathology at all. Then there were age-matched individuals without Alzheimer's and Alzheimer's patients. And what we look at here is an immunoprecipitation of the ryanodine receptor, and we look for what we call the biochemical signature of leaky channels, which includes pKa phosphorylation, cysteine nitrosylation. This is a measure of oxidation by detecting external carbonyl groups on the channel. So it would detect all oxidized proteins, but because this is an immunoprecipitation, it's specific for the ryanodine receptor. And we look at components of the ryanodine receptor macromolecular complex, including pKa, phosphodiesterase, PD4D3, phosphatases, and spinofilin, which is the targeting protein for PP1. And finally, this is the stabilizing subunit calstabin. So what's interesting is that there is an age-dependent oxidation of the ryanodine receptor, which we have reported previously in skeletal muscle. And there's also similar oxidation of the channel in Alzheimer's. But look at this, the channel in Alzheimer's is heavily pKa phosphorylated, and we don't see that in the non-Alzheimer's. And that's quantified in these bar graphs here. So we were interested in the possibility that pKa signaling targeting the ryanodine receptor could be playing a role in Alzheimer's. And we used a number of different mouse models, including this double transgenic APP PS1. And we've used 2 other mouse models, a triple transgenic as well, of familial Alzheimer's disease. And these show 2 different types of samples. One treated with one of the Rycals, S107, in the drinking water to prevent a leak in the ryanodine receptor. And these are mice, FAD mice crossed with a mouse that has a ryanodine receptor that cannot be pKa phosphorylated. These samples were then sent in a blinded fashion to the pathology department where they detected amyloid because these are human transgenes. This is human amyloid in a mouse brain. And both the pharmacologic and genetic treatment showed a dramatic reduction in amyloid deposition. And we also measured long-term potentiation, long-term depression, and both of those were impaired in the Alzheimer's disease mouse models and were improved by both the pharmacologic treatment and the genetic rescue at both 3 months and 6 months of age. This was pretty interesting because I think everybody would accept that one of the problems in Alzheimer's is impaired memory formation, but it had been less certain as to what the role might be of memory editing. And in this instance, we're showing that there's actually reduced erasure of memory or decreased editing. And this might contribute to the confusion in Alzheimer's patients. Not only can they not form new memories, but they can't edit the ones that they already have. And both of those were corrected by our therapeutic approaches. So we did a host of behavioral studies, and remarkably, while we were doing these studies, I had a French scientist doing a sabbatical in the lab, and we started presenting these data at lab meeting. He goes, oh, I have a colleague in Nice who's doing exactly the same thing. In fact, he bought S107 from one of the 13 companies that's now selling it illegally, breaking Columbia's patents, but I'm very happy that it's widely available. And what they had done was essentially a parallel study in 2 different mouse models, and we converged all of our data together because they were remarkably similar, and decided to join forces rather than compete. And so what we're looking at here is fear conditioning, where both contextual and cued fear conditioning, which was impaired in the Alzheimer's mouse models and improved both by the pharmacologic and genetic treatment. This is a novel object recognition test in which the Alzheimer's mice have decreased discrimination between the new and the old objects, and that was improved with both the pharmacologic and genetic treatment. The Morris water maze was partially improved in terms of finding the hidden platform with the drug treatment, and the rescue was more complete with the genetic treatment. And the elevated plus maze, the Alzheimer's mice tend to go to the open arms more than the wild type inappropriately, and that was also improved with the treatment. And so we come up with a model that, of course, needs a lot more exploration. But the idea here is that amyloid has been shown to activate beta-2 adrenergic receptors, and one of the targets could be pKa phosphorylation of the ryanodine receptor. Also, oxidative overload causing oxidation of the channel, a calcium leak that activates a number of calcium-dependent enzymes in the amyloid metabolic pathway, creating a vicious cycle and explaining why blocking the leak in the ryanodine receptor or preventing its phosphorylation could potentially impair some of the pathology in Alzheimer's. So we have a lot more work to do on this, but I wanted to share that story with you. I'm going to go back to the heart now and point out again that the biochemical signature of leaky channels is shown here with, in this case, phosphorylation, decreased amount of the stabilizing subunit. And when you treat with the Rycal drug, you can improve the binding of the stabilizing subunit. And in vivo models of heart failure in the mouse, treatment with the Rycal drug does improve cardiac function, measured here with an echo or a catheter in the heart. But in the mice lacking the stabilizing protein, has no effect, providing support for the proposed mechanism of action. So we started a collaboration with the French pharmaceutical company Servier about 10 years ago and developed that molecule that I showed you in that slide a few minutes ago, and it went through safety studies very nicely. And we did a phase 2 clinical trial in patients with severe heart failure using the doses shown here. Those are the numbers. Treated the patients for 12 weeks and measured the cardiac function with an echo looking at the ejection fraction. And you can see that there was a trend towards improvement. Now, the drug was not very potent, and there's no dose response here, which lessens the enthusiasm, but there was a pharmacologic effect, which is encouraging. And we also looked at a marker for heart failure called the pro-brain natriuretic peptide, and that's usually elevated in heart failure. And you can see at the different doses we studied in these patients that there was a reduction in that marker for heart failure. And finally, we looked at arrhythmias, which we were able to show the drug had an antiarrhythmic effect in animal models. And here, in a dose-dependent manner, it reduced the incidence of ventricular tachycardia in patients with heart disease. So based on these encouraging studies, we now are developing a second generation, much more potent form of the drug, Rycal, and that's now in phase 1 studies, which will be completed in April. So the idea then is a novel approach to treating these disorders, which is a small molecule that binds to the ion channel, in this case ryanodine receptor, doesn't affect the pore of the channel, but rather prevents the loss of an associated protein, which now stabilizes the channel function, decreasing the leak of calcium, but does not impair the normal activity of the channel. So all other known ion channel modulatory drugs, or most of them that I'm aware of anyway, either block or activate a channel and have a lot of side effects. These drugs seem to be well tolerated, and in clinical studies, they improve the function of the heart, ejection fraction. I didn't show you these data, but reduce the size of the heart, which is a good thing in heart failure treatment, and decrease that marker for heart failure. And those are phase 2A trials. Second generation Rycal, called ARM-210, started phase 1 in September, and that should be completed in April. And as I'll show you in a moment, on the basis of preclinical studies, we are going to progress to studies in Duchenne muscular dystrophy. So in skeletal muscle, a fascinating unresolved question has been what causes muscle fatigue. And the field of skeletal muscle was dominated for about a century by two giants, A.V. Hill and Otto Meyerhoff, who shared the Nobel Prize for Physiology or Medicine in 1922. A.V. Hill was working with a skeletal muscle preparation on the lab bench, repeatedly stimulating the muscle, and recorded an accumulation of lactic acid, which he reported was the cause of muscle fatigue. And this is why your mother has told you when you went out and exercised and you got burning in your muscles, she would tell you, oh, it's a buildup of lactic acid. And in fact, more recently, studies by Håkan Westerblad and others have shown that not only is the lactic acid not the cause of muscle fatigue, it's probably a metabolite that the muscle uses, but in fact, there is a decrease in the calcium signal for muscle contraction. So this caught our attention. And I was at a muscle meeting a number of years ago, bumped into a scientist who I was talking to, and he said, well, we have muscle biopsy samples from trained athletes. And he turned out he was at Appalachian State University, and he had all these world-class cyclists coming there, and they were letting him biopsy them. And I said, well, why are they coming to Appalachian State University? And it turned out Lance Armstrong spent a couple of weeks there one summer before he got discredited. So all the cyclists in the United States for a period of time would go there and train. And so what he had was trained cyclists working, able to do 70% max VO2 for 3 hours a day for 3 days. And he had before and after biopsies, which he sent us. And what we found was an increase in the PK phosphorylation after this extreme exercise, nitrosylation of the channel, and a depletion of the stabilizing subunit in these trained athletes. And so again, we term this the biochemical signature of leaky channels, and we think that the physiologic role may be to serve as a pop-off valve to prevent intracellular calcium overload, which could be toxic. But the key thing to think about is that these changes are transient. And so when you stop exercising, the leak goes away and you go back to a normal situation. Whereas in a disease like muscular dystrophy, Duchenne muscular dystrophy, the stimulus is chronic, as I'll show you. So Duchenne muscular dystrophy is the most common mutation in live male births, 1 out of 3,500. And despite discovery of the gene over 2 decades ago, the boys are still treated with steroids, wheelchair bound in their teenage years, and many die prematurely in their 20s. And so we became interested in trying to understand whether or not there might be a role for leaky ryanodine receptors, once again because of a long literature implicating calcium abnormalities with Duchenne muscular dystrophy. And what emerged from these studies that indeed, in probably a physiological attempt to increase blood flow to the damaged muscle and increase muscle function, there's a dramatic upregulation of the inducible form of nitric oxide synthase in dystrophic muscle. And one of the unintended consequences is the heavy nitrosylation of the ryanodine receptor, dissociation of the stabilizing subunit, a calcium leak which activates the calcium-dependent protease calpain, contributing to muscle damage. And we were able to show that treatment with the Rycal prevented the loss of the stabilizing subunit. And in studies that we did that have been corroborated in 2 independent labs, one in Italy and one in Washington, D.C., we showed in this very simple test where you take a mouse, put it in a cage with a running wheel. As soon as the lights go off at night, the mice run, take a drink of water where we put the drug, and then they jump on the wheel. And wild-type mice will exercise about 6 to 8 kilometers a night. And the dystrophic mice, about half that. And in a dose-dependent manner with the Rycal, we were able to improve this voluntary exercise. So I want to finish up the last few slides with a more personal story. And this has to do with a disorder called RyR1 myopathy. So there are very rare instances of individuals who have mutations in the ryanodine receptor, RyR1, the structure of which I showed you earlier in this talk. They develop a classic pathology called central core disease, which is simply named for the fact that their muscle has this white appearance in the center of the muscle. Nobody knows why. The phenotype is incredibly variable. There are congenital lethal phenotypes and all the way to almost unaffected. But the classic phenotype is a large proximal muscle weakness such that the individual cannot stand up from this position and they have to sort of push themselves up as they go. So I was contacted about 1.5 years ago by an individual who's a 40-year-old physician, has a known mutation in his RyR1. And at the time I met him, was very weak in his lower extremities. And in fact, when you'd be talking to him at a table, he would appear pretty normal. At the end of the meeting, his wife would come hook her arms under his armpits and have to lift him up. And so we genotyped his ryanodine receptor and found that he had 3 sequence variants, and we also genotyped his parents. And from his mother, he inherited 2 sequence variants in exon 34. One is a leucine deletion, another is an arginine to cysteine mutation. And from his father, he inherited this threonine to methionine. And where are these located? So looking down on the channel, you can see that the 2 exon 34 variants are right near where calstabin binds, and the exon 96 variant is near the port of the channel. Looking at the side view, you can see that the threonine to methionine missense substitution, which he inherited from his father, is right in the pore of the channel. And the two that he got from his mother on exon 34 are located in a very interesting portion of the molecule where there's enormous flexibility between the core of the molecule and the outer part of the cytosolic shell. So we looked at the biochemistry of his channel, and just to remind you, he told us he has profound muscle weakness in his quadriceps, but at that time he reported that his upper limbs were essentially normal. And so he asked me if we would do a biopsy, and I was extremely reluctant to do this because I thought one of the things that Jerry taught me when he was my professor at Harvard Medical School was never do a test on a patient unless you think it's going to make a difference in their care. And I really couldn't think of how we could help him with a muscle biopsy, but he was very insistent. I think I mentioned that he's a physician, so a little bit of knowledge can be dangerous. In any case, I arranged for him to have a muscle biopsy. Not that I would do it myself. I've grown up since those days. And we looked here at the biochemistry of the channel, and what we saw was indeed that biochemical signature of leaky ryanodine receptor in his affected muscle, but not so much in the unaffected deltoid of his arm. And there was, in fact, depletion of the stabilizing subunit. And when we incubated his muscle lysate with two different Rycal drugs, S107 and ARM36, we were able to show rebinding of the stabilizing subunit, suggesting that we were, in fact, fixing the leak in the channel. So we still had no idea which of these three sequence variants might be contributing to his phenotype, so we made recombinant channels with all the different combinations of the different sequence variants, reconstituted them into planar lipid bilayer, and studied their single channel activity. And only the combination of the two variants that he inherited from his mom showed an abnormal function of the channel. In this case, increased open probability under conditions where a normal channel would be tightly closed. And that's represented here in these bar graphs. So this suggested that potentially those two sequence variants in exon 34 could be disease-causing. And we looked at a microsomal leak assay, and indeed the leak of calcium, intracellular leak, was increased, and we could improve that with the treatment with the drug. So where are we taking this? Well, it's interesting once again to note that the potential Rycal binding site represented by this green peptide identified by ligand binding studies adjacent to the stabilizing subunit surrounds the two mutations that we showed may be causing a leak in this channel, potentially interfering with a critical part of the channel required for keeping the channel tightly closed. Well, my fellow in my lab, Alice Kushner, who's a cardiology fellow now, has gone into the literature and mapped the location of about 183 known RyR1 mutations. And you can see they're spread all throughout the channel. And we now are in the process of collecting clinical information and muscle biopsies on a number of these patients from colleagues around the world who take care of the patients, and will be able to see whether this N of 1 experiment that I've described to you today is held throughout. And in addition, Kate Muller at the NIH, who studies RyR1 myopathy, has 80 patients with known mutations. And if our drug gets through phase 1 safety trials, which we'll know by April, we're planning to do a clinical trial in these patients. So I just want to finish up by I've mentioned my collaborators in the structural biology. These are current members of my laboratory. Much of the work that I've described, biochemistry, has been done by Stephen Ryken. A lot of the physiological experiments by Gaetano Santulli, molecular biology by Aneta Ronska. Single channel studies were done by Chi Yuan. And I had mentioned Ron Zock as part of the structure project. And all the behavioral studies and the brain studies were done by Xiaoping Liu, who worked closely with Mike Shalansky's group. This is a black-tipped reef shark in Fiji, which came right up to my dome port of my camera. I thought I got a really good shot. People look at that and think I'm insane. And this little critter up here is Hippocampus bargabanti. It's one of 7 known species of pygmy seahorses, which were unknown to exist until 2007. And interestingly enough, the satellite that the North Koreans fired, which orbited over the Super Bowl, before it got there, the rocket ship that fired it off dropped into the ocean right on top of where Hippocampus bargabanti lives off the coast of the Philippines. So there are consequences, and I noticed that the Simons Foundation has a program in ocean ecology, and so I thought I would show a couple of pictures. So thank you very much for your attention, and glad to take questions.